Hey everyone, this is Liam Sanyo from Inside Scientific, and in this special episode of Industry Insights, we're meeting with 3 immuno-oncology experts at Crown Bioscience, Dr. Saskia De Man, Dr. Marrit Putker, and Dr. Pirouz Daftarian, who recently joined us for a webinar about the impact of T-cell directed immunotherapy on the cancer immunity cycle. In case you missed the webinar, you can find a link to it in the description here, but today we're going to be taking a deeper dive into some of the concepts and addressing some of the best questions we were asked. Okay then, Saskia, Marrit, and Pirouz, thanks so much for joining me today. Are you ready to answer a few questions here?
Of course.
Of course. Thanks.
Fantastic. Well, first of all, we got some great feedback from our audience on the polls. Was there anything that stood out to you?
Yeah, thanks, Liam, for that introduction. So the first poll we showed during the webinar was on what kind of assays is our audience using to evaluate their T-cell therapeutics. And what really stood out to me was that a large percentage of the responses replied that they focus still on cytokine analysis, as well as, for example, flow cytometry readouts. In the webinar, we also described our high-content imaging platform, and I wanted to share a bit about how that is different and what that can add compared to those FACS and cytokine readouts, even though they are very useful to study, for example, the activation of the immune cells in any study, in any assay. One thing that we think is really a benefit of the high-content imaging platform is that we can also really assess the effector function. So not only are these immune cells activated, but also are they migrating, are they infiltrating tumor objects, and can they kill those tumor objects. And one of the great things is that also at Crown Bioscience, we can combine these different readouts. So at the end of our high-content imaging assay, it's always possible to also collect the supernatants, and some of our biomarker teams also can do the analysis on that. But really to have not only the activation status, but the effector function can be very useful, I think, in many research questions. So that really stood out to me on the first poll, and maybe Marrit can give some insight into the second poll.
Definitely. I think one thing just to add, actually, Saskia, to what you were just saying is that also it's very interesting to review those different parameters because they don't always align, right? So whenever you find an increase in your cytokine profile, it doesn't always mean that you also see increase in killing.
Exactly.
So I think the fact that we can distinguish those two readouts is actually a nice added value for this.
I think we'll talk a bit later about our checkpoint inhibitor assays. But there, for example, even though we see large increases in from gamma production, signaling activated immune cells, we don't see enhanced killing effects. And so that is a discrepancy that is indeed interesting to measure with the different techniques.
Yeah, and if we then move on to the second poll that we also had, which was the question was, what cancer indications are of interest to you? We had a whole list of cancer indications listed there: brain, breast, lung, liver, pancreatic, gastrointestinal, kidney, prostate, cervix, and other was the last answer. And what was nicely indicated is actually there's a very nice spread in our audience in different fields of interest. Very clear enrichment for the classical lung and breast cancer interest and gastrointestinal interest. But overall, I think we actually see interest in most of those indications. We also received questions about this in our chats afterwards regarding which models we actually have available as Crown and for which assays. And it's safe to say for the in vitro platform that we have availability for all of the epithelial tissues. For the ex vivo platform, we also would have probably brain tumors, but for the organoid platform, we only have models available from epithelial tissues, which is kind of everything I just named without the brain organoids, which is something we may be interested in developing, but it's really from a different technical aspect, because the organoid models that we've been describing so far, they're all based on epithelial stem cells, hence also this focus on those type of tissues. And then from there, for all of those indications that we have available, so those are over 17 tumor indications now available, we will be able to, all of the assays that were described that we are doing, that we're offering as organoid surface, we can use all of those different indications in those assays. So, for example, all of the co-culture model, all of the co-culture assays that Saskia has been describing during the webinar are available. Most of the indications have already been tested, and some of them will still need to be optimized. But so far, all of them have been successful, so I don't see any problems with that. Then on top of that, we of course we can do so the simple co-cultures, but also the reconstituted TME assays. We have all of those organoid models available, where we can combine them with either immune cells, but also, for example, with CAFs and myeloid cells. So these type of co-cultures will also be mentioned and actually discussed in depth in our next webinar. And lastly, there's the more autologous setups that we have. So that's one is the setup that I described during the webinar, which is talking about the colorectal co-cultures, where we're developing the autologous setup. So deriving the TILs or the PBMCs from the same patient and putting them into a co-culture. But also we have this ex vivo patient tissue platform, where we're deriving directly from the tumor the tumoroids and seeding them into the plate. So really with primary tissue running drug screens. And there, of course, because it's so primary, it's also autologous setup. So all of the autologous TME components are in those cultures, allowing actually to look more into the autologous responses.
Yeah, and maybe I can touch on that as well, because we also saw that question popping up, like which IO assay would be best to use. Of course, it depends very much on your question. And one of the things is this difference between autologous and allogeneic systems that is often asked also in a day-to-day basis to us. And I just wanted to touch upon what we can do with both systems. And I would like to start with the allogeneic setup. And one thing is that this also comes with a lot of benefits. So sometimes clients are a bit hesitant to start with that. But what we see is that it's very scalable. So we can really run many conditions, many plates, because we're working with healthy donors. So you get, for the blood, you get large amounts of cells, which allows large testing capacity as well. And also for many treatment modalities, we really see very good assay windows. So, for example, with antibody-dependent cell cytotoxicity, or for bispecific antibodies, or for cellular therapies, or things like that, or other immune modulators that target, for example, the myeloid cell compartment, we really get really nice results in those allogeneic setups. And so there's and that really allows this scalability. And indeed, the innate tumor microenvironment and the autologous system can be very important for some drugs as well, right? And this will be discussed more in the next webinar. But it also comes at some cost. So, for example, there the scalability and the reproducibility is more difficult because you are limited in the material you can get from one patient, right? And so that really depends on your question. I think one treatment modality that I left out of my list just now is checkpoint inhibitors. And there we really see that in the conventional setup of the allogenic system, it's very hard to observe any responses on killing. And there, for example, in the primary tissue platform, we really do see similar response rates as we also do in the clinic. So about 20%, depending on the indication. And also we think that with optimizing the autologous system with organoids that Marrit discussed in the previous webinar, we might also be able to test that at some point. But of course, Pirouz also joined us in the webinar, and also getting allogenic PBMCs into an exhausted state might help us get a bit closer to that exhausted phenotype that is also correlating with checkpoint inhibitor reactions. So yeah, that's a bit the difference between autologous and allogenic systems that I wanted to touch upon.
Yeah, fantastic. Thanks so much, Marrit and Saskia, so far for the great insights. Covered a lot of ground between, the polls, models, and assays available, autologous versus, allogenic systems, checkpoint inhibitors. But Saskia, I think what you just said leads into a nice question. And Pirouz, I think you can cover this. Is Crown Bioscience's T-cell dysfunction and exhaustion assay ready for screening or characterization of immunomodulatory drug candidates?
Thank you. The assay that I described in the webinar is ready in this sense that we have few rounds of activation or stimulation of T cells as it happens in the tumor deposit to model it. And as it happens in the tumor deposit, we have in our setting the upregulation of co-inhibitory molecules as a positive feedback, as well as we have the inhibition or suppression of functional parameters such as key cytokine measurement like interferon gamma, TNF alpha. And also on top of that, because like in other platform, also you saw that the killing is one of the verdict of in these kind of in vitro setting. We also show that the killing has been compromised or suppressed. So with that sense, the assay has been repeatedly, reproducibly showing the same in different donors that we have tested and validated the assay. Yes, the assay is validated with that sense. In terms of reversing or curing these T-cell exhaustion, this is the stage we are at right now. And yes, we will be happy to include drug candidate and for this phase of the T-cell dysfunction, T-cell exhaustion assay. The readout that we are right now conducting, although we can some other readouts are possible, but to focus on the main major telling readouts in our assay, we are measuring cytokine. We're measuring this like immunophenotyping of the co-inhibitory molecules, and we also conducting a killing assay to show the killing effect or impact of the drug candidate on reversing the lost killing ability of these exhausted or dysfunction T cells. What I want to just add is that this assay also can be translated to CAR T cell because that's the same scenario that you have repeated or persistent of the antigen. So that's also doable. Thank you.
Yeah, thank you, Pirouz. It's a great answer. I think that's just about all we have time for today. So thanks again, Pirouz, Marrit, and Saskia for being with us today. That was great.
Thank you.
Thank you.
Thanks everyone for joining us today. If you enjoyed this episode, be sure to check out our recent webinar together. You'll also be able to catch the next webinar in our series starting January 26th, which will be about analyzing the suppressive tumor microenvironment using in vitro assays with an accompanying podcast published shortly after that. We hope to see you there, and have a great day, everyone.